Good afternoon, everyone. I am Dr. Atima, DGM Marketing of Dr. Lal Path Labs, and will be moderating the session of the day. So before we begin the session, I would like to introduce Genewise. Genewise is the new genetic testing division of Dr. Lal Path Labs with a focused foray into the genetic testing. With the advent of this division, all of you can expect many revolutionary and fast-making novel tests leveraging the latest technologies, resulting in faster turnaround time and quality reports at the most affordable prices for your patients. Genewise offers a wide range of key tests related to oncogenomics, neurogenomics, rare diseases, and prenatal, postnatal genetic testing using cutting-edge technologies such as NGS, MLPA, ddPCR, FISH, and Sanger sequencing. All these technologies are being performed in-house at our National Reference Lab. As we are all aware, the genomic space is dynamically changing, and to keep up with the pace and remain the market leader, we wish to take the genetic testing from biology to health and from health to society. This is a snapshot of our core technical team of National Reference Laboratory, which includes Dr. Atul, who is heading the molecular department, performing the tests like exomes, SCAR panels, neuromuscular panels, Dr. Rajiv as our histo head, Dr. Anil as the HOD hemat, who performs tests like ACHR, MUSK, Dr. Jasmeet, the immunology head, Dr. Saurabh, who heads the cytogenetics department and performs microarray, including HD microarray, exome microarray, and Dr. Beena handling the flow department. Next, I would like to introduce the speakers for this session as to go so that we have no interruption in between the talk. Our first speaker is Professor Ghosh. He is an MBBS, MD, DND, FICOG in Advanced Fetal. He is the Director of Institute of Fetal Medicine, Kolkata, and a President of Society of Fetal Medicine, Kolkata. And he has been a past President of Bengal Ops and Gynae Society. Our second speaker would be Dr. Saurabh. Dr. Saurabh is a PhD in clinical cytogenetics from Vellore, VIT University Vellore, and he is heading our department of cytogenetics. And he has successfully implemented and practiced a novel cytology of ICP, interface chromosome profiling, for the first time in India. Our third speaker is Dr. Leena. She is a molecular geneticist with 10 years of research experience and is currently working as senior scientist in the Department of Cytogenetics. Let's now move to the academic treat by Dr. Ghosh. The questions can be typed in the chat box, and we will be taking it at the end of the session. Over to Dr. Ghosh. So please, you can...
At the screen. Can you share? Can you see the screen?
Yes, sir, it's visible.
Okay. Thank you very much for the kind introduction. Good afternoon. And I'm really honored and grateful to Lal Path Lab for inviting me to talk on a very important topic, genetics of recurrent pregnancy loss. We do talk of recurrent pregnancy loss in our gynecological forum a lot of times, but hardly do we actually discuss about the genetic aspects of recurrent pregnancy loss. And this is a very evolving area. It's very fascinating as well. And Lal Path has really gone leaps and bounds into this arena and has come up with quite a new innovations. But first of all, we have to come for the scientific and see what is the role of genetic testing of recurrent pregnancy loss. And good afternoon from Kolkata. Now, whenever we talk of genetics, it really first thing that hits us is for the busy practitioner that as ACOG 2016 has really commented that genetics and genomics is viewed as a very complex nuisance that interferes with the clinical management. Oh my gosh, do I have to read genetics again? Why have I come to obstetrics and gynecology then? And I don't have very little time to focus on real issues. I'm so interested in delivering babies and taking out uteruses. But to the uninitiated and for our satisfaction, it's good to know that genetics is vast, but what we need to know is only a very small size of that apple. The human journey begins as very tiny microscopic gametes, the egg and the spermatozoa, which teeming millions come in and basically enter one egg, and the entry of the other sperms are blocked, and the zygote is formed and the cleaving embryo starts. How all these things are done, we really don't know. They are definitely pre-programmed, and that's why it's a role of genetics in a very big way. And therefore, implantation and maintenance of pregnancy is really a wonder of nature. And whenever there is any derangement of this mechanism, it results in pregnancy loss, which brings to our subject of recurrent pregnancy loss. How do I go to the next slide? Can't go to the next slide. Okay, so pregnancy loss is a spectrum. All pregnancy losses, as we call pregnancy loss, from the conception till 24 weeks of gestation, which accounts for about 13.5% of all conception. Can you hear me? Hello? Can you hear me?
Yes, sir. Yes, sir.
Okay.
We can hear you loud and clear.
Okay, the term miscarriage is better avoided. But of course, there is pregnancy loss even beyond 24 weeks, which results in intrauterine stillbirth and premature labor. But we are going to take those at a later date, maybe, but not today. Pregnancy losses can occur in first trimester, in early second trimester, between 12 and 24 weeks. And there is a third category, which is called a biochemical pregnancy loss, before even the patient loses or misses a period. And the recurrent pregnancy loss, as we see, is ideally or really the tip of the iceberg, because at conception, only 30% of all the embryos which conceive actually get implanted. And after implantation, almost 30% of them get miscarried. And then it comes to the clinical miscarriage. Clinical miscarriage is after the woman misses her period, and that accounts for around 10 to 15%. And live birth is only 30% of the conceptions. So 70% of all zygotes eventually are discarded before even delivery. And this is probably a nature's method of natural selection. So we can divide miscarriages into preclinical and clinical. What we're going to deal today is the clinical portion. There is a very interesting jargon called the recurrence of recurrent pregnancy loss. If in each pregnancy, we know there is a pregnancy loss of around 15% or so. After miscarriage, it increases to around 20%. After 2 consecutive miscarriages, it increases to around 30%. And after 3 consecutive miscarriages, it increases to around 40%. So this is very interesting to say that recurrent pregnancy loss does not occur by chance. If it was by chance, then the chance of third loss would have been only 0.33%. Like the chance of one pregnancy loss, as we said, was 15%. And the second loss would be 15% of that 15%, which would be 2.25%. And the third pregnancy loss would be 15% of that 2.25, which would be 0.33%. But in actual real life scenario, we see that the incidence of recurrent pregnancy loss is almost as high as 2 to 3% and in different papers. So obviously this cannot occur by chance. There must be some mechanism which is predisposing some women to habitually abort, and this is not a barn door natural population. It all started in 2011 when the Royal College of Obstetricians and Gynecologists came up with a green top guideline number 17 in 2011, where it was titled Investigation and Treatment of Couples with Recurrent First Trimester and Second Trimester Miscarriages. But a lot of water has flown under the bridge after that. And the one which we follow most often these days is the ASRM, the American Society of Reproductive Medicine Practice Guideline Committee Opinion of 2012, where they have considered only the clinical pregnancies. And recurrent pregnancy loss has been defined as more than equal to failed clinical pregnancies documented by ultrasonography or by histology. So biochemical pregnancy is excluded, and it also excludes ectopic pregnancy which are not visualized, and it also excludes spontaneously resolved pregnancy of unknown location, which we call PUL, or persisting pregnancy of unknown location, which is called PPUL. So pregnancy loss suggestive of miscarriage but not documented by ultrasonography, and where the tissue is not collected for documentation, is not calculated as recurrent pregnancy loss. That's an interesting advancement that we are today. Again, the American College has gone on to divide recurrent pregnancy loss into early recurrent pregnancy loss, where there are 2 or more intrauterine pregnancy losses less than 10 weeks, and recurrent pregnancy loss of more than 2 or more intrauterine pregnancy above 10 weeks. Now, why is this? Because it has been recognized that generally the causes of pregnancy loss below 10 weeks are a little different from the causes of pregnancy loss after 10 weeks. Like below 10 weeks, we have mostly genetic reasons for loss and hormonal reasons for loss, mainly luteal phase defect and progesterone deficiency. Now, when we go to more than 10 weeks loss, women who are habitually aborting after 10 to 12 weeks, mostly they are because of uterine or anatomic problems like subseptate uterus or cervical incompetence, submucous fibroid, because of infections, because of bacterial vaginosis, etc., immunological mechanisms, and APLA syndrome, antiphospholipid syndrome. So the spectrum of pregnancy losses are quite different. Now, what we are interested in are the recurrent causes of recurrent pregnancy loss. Causes of pregnancy loss and causes of recurrent pregnancy losses are different. Like the American College has again recognized that there are different causes of recurrent pregnancy loss: genetic, anatomic, endocrine, immunological, and infections. Now, for today's purposes, we will concentrate on genetic causes of pregnancy loss, which accounts for only 5%. But this would be an underestimate, because a lot of these idiopathic pregnancy losses, recurrent pregnancy losses, and immunological pregnancy losses have a genetic background. So the exact component of genetic reason for recurrent pregnancy loss is more than what meets the eye. And very interestingly, there is an overlap of spectrum between recurrent pregnancy loss and infertility. For example, in infertility, we have causes like Y microdeletions, azoospermia, antisperm antibodies, immunoglobulin, etc., which have genetic background. And in recurrent pregnancy loss, we have chromosomal arrangements, antiphospholipid syndrome. Antiphospholipid syndrome also has some genetic background. And of course, there are common factors like aneuploidy and chromosomal aberrations like CMA abnormalities, microarray, and genetic like exome sequencing, which can also cause infertility and recurrent pregnancy loss. So there is quite a bit of overlap between the two. So it is sometimes thought there might be reasons which cause infertility, and they carry on into later pregnancies to cause recurrent pregnancy loss. And then ultimately, if they survive, the fetuses survive, they can have some congenital malformations. And after that, after delivery, they could be having some neurobehavioral abnormality, even if they're not having any phenotypic abnormality. So it's a whole spectrum of which we are only scratching the surface. There is another very interesting association between age of the mother, spontaneous abortions, and fertility. As the age of the mother increases, the fertility decreases and the incidence of abortion increases. There is a genetic background there as well. As this study, which was this year, published only a few months ago, shows there is a high incidence of diminished ovarian reserve in young, unexplained recurrent pregnancy loss patients. And women, we all know, with decreased ovarian reserve have high resistance eggs, which are in some way not quite normal, deranged, and might be having some genetic or chromosomal defects. And they can cause infertility as well as recurrent pregnancy loss. Now, when we talk about workup of recurrent pregnancy loss, the ASRM guideline 2012 is more or less the thing which we all gynecologists follow, and which goes on to say that there is an euploid POC, product of conception after 2 pregnancy loss, or at least 2 consecutive miscarriages with no product of conception diagnosis, or at least 3 non-consecutive miscarriages with no product of conception diagnosis. We have to evaluate anatomical, endocrinological, progestogen, LPD, autoimmune factors like APLA, evaluation of lifestyle, environment like caffeine, tobacco, etc., and of course, a part of it last is the genetics. And what do we mean by genetics of recurrent pregnancy loss? This is something which is very interesting, because to all gynecologists, traditionally, genetics starts and ends with karyotyping. And we all know that, yes, in recurrent pregnancy loss, we have to send the product for karyotyping, and we have to send the parents for karyotyping. And that is where our funda ends. And if we do send these things for karyotyping, either parental blood or the product of conception, in the product of conception, what we are likely to see are polyploidy. Yes, of course, we can see euploidy and haploidy, but more commonly polyploidy, which is triploidy or tetraploidy, and aneuploidy, where the chromosomal number is 1 more or 1 less, which can affect the autosome in the form of trisomy, where there is 1 more, the commoner ones being Patau syndrome, Edwards syndrome, and Down syndrome. Monosomy of autosomes is lethal, which cannot persist and are definitely aborted. Sex chromosomal abnormalities like trisomies, like the Klinefelter, XXX syndrome, and the XYY syndrome, and monosomy of chromosomal sex chromosome, which is classically the Turner, or it could be mosaic Turner. Now, abnormalities in structure in the karyotype is more commonly seen in the parental karyotyping, not so much in the fetus, but yes, it can be seen. Translocations in the parental karyotyping could be balanced or unbalanced. There could be deletions and there could be inversions. So these are the different types of karyotypic abnormalities which we know more or less. Now, going a little bit more into parental chromosomal arrangements, it says that the American Society, that in approximately 2 to 5% of couples with recurrent miscarriage, one of the partners carries a balanced structural chromosomal anomaly, and most commonly a balanced reciprocal or a Robertsonian translocation. That is, one part of chromosome goes and gets translocated into another one. And although carriers of a balanced translocation are absolutely phenotypic and normal like you and me, their pregnancies are at increased risk of miscarriage and may result in a live birth with, as I said, multiple congenital malformations or neurobehavioral abnormality secondary to an unbalanced chromosomal arrangement. So the balanced chromosomal abnormality in the parent, which is phenotypically normal, can result in an unbalanced chromosomal arrangement in the product of conception, which can cause anatomical abnormality or behavioral abnormality. And the risk of miscarriage is influenced by the size and the genetic content of the rearranged chromosomal segments, deletions or duplications most commonly. These are translocations, which I'm sure our next speaker, Dr. Saurabh Bhattacharya, is going to go in details about Robertsonian translocations and balanced reciprocal translocations. Now, for example, we do send blood for parental karyotyping, and the report comes as translocation. So what is the risk of subsequent miscarriage? If there is a reciprocal translocation, the risk of miscarriage is around 50 to 70%. If there is Robertsonian translocation, it's slightly lower, 30 to 50%. If there are inversions, or it is about 30%, depends on the amount of inversion and deletion, duplication, and normal is around 30%. Now, if we come to the embryonal chromosomal abnormality, in couples with recurrent miscarriage, chromosomal abnormalities of the embryo account for about 30 to 57% of further miscarriages. The risk of miscarriage resulting from chromosomal abnormalities of the embryo increases with advancing maternal age, as we discussed before. However, it is important to note that as the number of miscarriages increases, the risk of euploid pregnancy loss increases. Now, if we analyze the chromosomal abnormalities by microarray in the product of conception of embryos, which has been done in this study 2014 by applying PGD, we see that the common chromosomal abnormalities causing recurrent miscarriages are chromosome 13, chromosome 2, and chromosome 9. One which we talk of aneuploidy so much, like Down syndrome, accounts less. And recently, we come to the recurrent pregnancy loss guideline, which was published by ESHRE, the European Society for Human Reproduction and Embryology. This is published in 2017, and they have almost replicated the one which we discussed about the American Society for Reproductive Medicine. And the genetic test which has been circled here is definitely one of the things which needs to be done, which we need to know is when to do it and what to do it. So recurrent pregnancy loss, as far as genetic workup is concerned, depends on whether the chromosomal pattern of product of conception is known. For example, she has had a miscarriage and somebody has bothered to pick up that product of conception and send it for genetic analysis. Or there are some instances where the patient would have had a miscarriage or abortion or a pregnancy loss and the product of conception has gone down the drain. In that case, we don't have the chromosomal pattern of the product of conception. Now, where the product of conception chromosomal pattern is known, then if she has a second consecutive miscarriage and third non-consecutive miscarriages, we need to do a fetal product of conception. From here, the ball starts rolling. So if we know the fetal product of conception, then it could be an euploid karyotype, which means that the product was actually chromosomally normal. If the product was chromosomally normal, then there is more than likely that the parents are okay. And we have to then concentrate on the other causes of recurrent pregnancy loss workup, which we have discussed before in the American Society guideline as well as the ESHRE guideline. But if the fetal product of conception, which normally we used to talk about karyotype, I will restrict myself to karyotype, particularly in this slide, it shows an unbalanced chromosomal translocation or inversion in the product of conception, then we need to do parental karyotype and offer pre-implantation genetic diagnosis or gamete transfer, gamete donation or adoption or things like that, ICSI. So these are for future, these are the options that we have. Now, when the product of conception is not known, like it has gone down the drain, then we have no other option than to straight away go for recurrent pregnancy loss workup in the way that American Society has done. And if the parents, if we don't know, then it is ideally better to go for parental karyotyping and take it from there. Now there are nuances of karyotype, which means karyotype is slow, it is time taking, resolution is only up to 5 megabytes, human expertise is required for karyotype, and it most importantly, it needs live cells. Now in majority of cases in product of conception, the cells are already dead by the time the product reaches the lab. And sometimes even we don't know how to send it, and some of us send it in formal saline, which is really a shame. It should be sent in normal saline or dry. So instead of doing karyotype, which sometimes fail, there is a method of only doing very quickly, a little cheaply, only looking for the normal, more common chromosomes like 13, 18, 21, 23, which is X and Y. And that is done by fluorescent in situ hybridization, which is called FISH. In FISH, what we've done is there are specific probes which are fluorescent dye labeled, tagged, and they get attached to specific sites of those chromosomes and can be identified under a fluorescent microscope. So the test, which is called FISH, looks for the presence or absence of that particular fluorescence in the target area of that chromosome. It is very specific. And there are two basically clinical uses of fish in recurrent pregnancy loss. One is to look for chromosome number, either to diagnose increased number or decreased number. And the presence of an extra fluorescent dot signifies an extra chromosome. And the other one is if we really know that the parents are suffering from or harboring a particular deletion or duplication, then we can diagnose the specific deletion disorder. We can make a probe for that particular area of the chromosome to see whether this baby or product of conception has a particular named syndrome or not. But this is very rare in modern obstetric practice. In the nuances of fish, if we discuss, we understand that we still need a microscope for fish. It is still cytogenetics, which needs cells, intact cells. At least 50 cells need to be analyzed to avoid error. It can be done on dead cells, but not autolyzed cells, but no cell culture is required. So that is one of the advantages of fish over karyotype, but we can only look for 5 or 6 chromosomes. Now, so far, we talked about cytogenetics. Now, these days, for recurrent pregnancy loss, we need to move a little forward and go into molecular genetics. Now, one of the things which we need to understand, because this term chromosomal microarray is going to come over and over again, and this is actually a form of molecular genetics. Now, if we open up a chromosome, it ends up in a long chain of double helical molecule, which is called DNA. It's almost about a foot long in each chromosome. And this DNA is the one which is very important and is the constituent structure of each and every chromosome. In karyotype, we look at the details of the chromosome by bending techniques and through microscope. But in chromosomal microarray, this can be done through computer at a very, very minute details, and you can interrogate all the chromosomes at a greater depth to look for gains or losses of nucleotides in any particular area of chromosome, which cannot be diagnosed by human eye. We have to have computers and bioinformatics system. Chromosomal microarray can be used as a low resolution or a high resolution, like whether we are using a high power telescope or whether we are using a low power binocular. So that these are the two differences between low resolution chromosomal microarray and high resolution microarray, which I'm sure Dr. Saurabh Bhattacharya is going to explain later on. Now, the important points to note about CMA are that the karyotype looks at a resolution of 5 megabytes, but CMA looks at 50 to 200 kilobytes, which is very, very high. And even if the karyotype is normal, and in those patients, if we do CMA in the same product, we see 8 to 13% more yield, which means the CMA will be abnormal where karyotype is normal. That's why chromosomal microarray is rapidly becoming the gold standard for product of conception analysis and not KT in cases of recurrent pregnancy loss. And there are a number of copy number variations which have been described in cases of chromosomal microarray from placental tissue in recurrent pregnancy loss, which is beyond me. I don't understand any of those. So I'll quickly skip that slide. Now, the important points to note about CMA is that CMA looks at clinically relevant deletions and duplications at a greater depth than karyotype. The recommendation is to do CMA of the product of conception rather than karyotype, which we said. There was a very interesting article in 2018 Human Reproduction. It says that recurrent pregnancy loss evaluation combined with 24 chromosome microarray, which means 22 autosomes and sex X or XY, mix of 24 chromosome microarray of miscarriage tissue provides a probable or definite cause of pregnancy loss in over 90% of patients. If we only restrict ourselves to the standard American College or ESHRE guideline, we are able to diagnose only about 50% of cases of recurrent pregnancy loss, and the rest 50% almost becomes idiopathic, which we don't know the cause. But if we use chromosomal microarray, then many of these cases we come to a diagnosis. So this paper goes on to say that recurrent pregnancy loss is estimated to occur in about 2 to 4% of reproductive age couples. A probable cause can be identified in approximately 50% of patients after an ASRM recommended workup guideline, which we discussed before. but over 90% of patients with recurrent pregnancy loss will have a probable or definitive cause identified when combining genetic testing of various sorts on miscarriage tissue with the standard ASRM evaluation for recurrent miscarriage. Very interesting article. And this year, again in October, this particular month, this paper has come out a few days ago, a new algorithm for the evaluation of recurrent pregnancy loss, redefining unexplained miscarriage. So the proportion of unexplained miscarriage is coming less and less, and is a review of the current guidelines. So whatever guidelines we have been following so far needs to be reviewed on the basis of these findings. And the paper is very interestingly shows that if we just do ASRM guideline, then fewer than half of the couple will be evaluated and will find the cause. If we do the recent guideline, then more than 90% of cases will be diagnosed in recurrent pregnancy loss, which emphasizes the genetic aspect of recurrent pregnancy loss. And I'm so happy that Lalpath has actually decided to do a webinar on this very interesting topic. A 24 chromosome CMA testing from the miscarriage tissue provides with an explanation of unexplained pregnancy loss will be reduced to less than 10%, which means more than 90% will find the cause. But of course, it has to be replicated. And they go on to say that incorporation of this strategy will result in significant cost savings to the healthcare system. Now let us move on from chromosomes to the genes. What are genes? A gene is a particular part of a DNA molecule that codes for a specific protein, and genes are made of DNA. As the picture shows, this portion of DNA is called a gene, and each chromosome contains many genes. There are approximately 1,000 genes per chromosome, which makes up around 23,000 genes in our every cell, which is called the human genome. And as far as recurrent pregnancy loss is concerned, lots of genes have been identified or reported as cases in when exome sequencing has been done, and the number of genes which are coming up as probable cause of recurrent pregnancy loss is increasing by the day, and is an endless thing. But what do these genes do? These genes are responsible, like for example, we say APLA. Now, why should antiphospholipid syndrome suddenly appear in some women and some women will be fine? This also has a genetic base. Factor V Leiden mutation, prothrombin mutation, factor 12 deficiency, hyperhomocystinemia, we all know are causes of recurrent pregnancy loss. And we used to test homocysteine and prothrombin and things like that. But they all have a genetic basis, protein S deficiency. So in near future, maybe we'll be testing genetics rather than the biochemical products and the hematological products. Now, coming back to the summary of recurrent pregnancy loss guideline by ESHRE, genetic analysis of pregnancy tissue is not routinely recommended, but it could be performed for explanatory purpose. For genetic analysis of pregnancy tissue, I'm explaining this in a minute. For genetic analysis of pregnancy tissue, array-based comparative genomic hybridization is recommended, which we have already discussed. And parental karyotyping is not routinely recommended in couples, depends on whether the product of conception is euploid or aneuploid. Now, therapeutic interventions after we have found out the genetic cause is already important. The first thing is these couple should receive proper genetic counseling. We gynecologists are not equipped enough to counsel such women or such couple. So a trained genetic counselor with laboratory specialists are required. And the options like adoption, gamete donation, ICSI, pre-implantation genetic diagnosis, all these things need to be discussed, which is beyond the scope of this presentation. There is another very important area of genetics, which is called epigenetics. Whereas not strictly genetic, changes in the human epigenome are increasing being associated with pregnancy loss and infertility. Epigenetic mechanisms include the way in which the genome is packed and thus the ability for the genes to be activated. And epigenetic changes can be inherited across cell divisions or across generations and can have a profound effect on individuals' phenotype. And there are a lot of work going on. regarding the epigenetic variations in, in the couple, which can cause recurrent pregnancy loss. So in a nutshell, ladies and gentlemen, this is the final slide which I want to present. And genetics of recurrent miscarriage and pregnancy loss, it shows that to date, research lines are focused on genetic and epigenetic polymorphisms related mainly to immune response and inflammatory mediators, and have yielded a significant relationship between recurrent miscarriage and immune mechanism. However, it is important to know that more evidence is required for them to be included in medical practice and obstetric guidelines. And if we can go back and if we see this one, the first slide here, that genetic analysis of pregnancy tissue is not routinely recommended till now, but maybe in near future this will become a armamentarium. So after we have spoken for about half an hour or so, 20 minutes, I don't know, what is the one slide summary of what we discussed so far? Because people might be really confused or what to do and what to not to do. So the next slide which comes is very important. And if you want, you can actually do a screen sharing of this. Basically depends on what to do regarding genetics of recurrent pregnancy loss as we stand today. If a woman has 2 or more miscarriages, we diagnose as a recurrent pregnancy loss. And ideally, we should be sending product of conception for chromosomal microarray, or if we don't have financial resources, at least for fish. We tend to avoid karyotyping because sometimes this karyotyping do not grow. But yes, of course, you can send it. Now, if the CMA is available, then the CMA could be normal or it could be abnormal. If the CMA is normal, then any form of problem in the parents, in the parents translocation, is unlikely. And therefore, we have to concentrate on the other causes of recurrent pregnancy loss, which we discussed other than genetic factors, which means these are non-genetic causes of recurrent pregnancy loss. If the CMA is abnormal, then we have to do parental karyotyping, because there is a possibility of balanced translocation in the parents who are phenotypically normal. And this is probably one of the very important indication for karyotype in modern obstetrics. Now, what happens on the right-hand side if the product of conception, CMA or fish or KT is unavailable? Then we don't have any other option than to go to parental karyotyping straight away. So this is in a nutshell what we discussed so far, ladies and gentlemen. Medicine is a science of uncertainty and an art of probability. In unraveling the mysteries of recurrent pregnancy loss, we have come a long way, but we still have a way to go. Thank you. Over to Saurabh.
Thanks a lot, Dr. Ghosh.
Yeah, thank you, Dr. Ghosh. Can you hear me?
So we'll manage this presentation from the deck.
Okay. So now over to Dr. Leena. She will take forward from genetic aspects of recurrent pregnancy losses, and then I'll discuss few interesting case studies. Over to Dr. Leena.
Good afternoon, everybody. Thank you, Dr. Ghosh, for a very enlightening presentation on the clinical findings of recurrent pregnancy losses. So I'll be discussing about the chromosomal aneuploidies that occur in the fetus and are associated with recurrent pregnancy loss. And along with that, I'll be discussing about the diagnostic perspectives leading to it. So recurrent pregnancy loss, as now we are all well aware of it, how does it happen? It is basically the 2 or more consecutive frequent pregnancy losses that occur in a woman prior to 20 weeks of the end of the last menstrual cycle. So it has been seen that approximately 30 to 50% of all the conceptions that occur, recurrent pregnancy loss is a common phenomenon. And 50% of it can be detected is basically because of the chromosomal abnormalities that occur. And it has been seen that approximately of the 15% of all the clinically recognized pregnancies, 45 end in recurrent pregnancy losses because of the advanced maternal age. The etiology of recurrent pregnancy loss is multifactorial, as Dr. Ghosh has already explained, that it can be because of the autoimmunological factors, it can be because of the endocrinological factors, anatomical factors. 40 to 50% are because of the idiopathic factors, and only 2 to 5% is because of the genetic factors. Genetic factors, it can be because of the parental chromosomal rearrangement that has taken place and is being transmitted to the future progeny, or it can be because of the de novo fetal aneuploidies. I will not be discussing the details of the cell cycle stages and mitosis and meiosis, but just to give you a brief on this is that the defects in the cell cycle regulation or the cell cycle checkpoints basically result in the recurrent pregnancy losses, as they hamper the chromosome structure, and this is then forwarded, transmitted to the further future progeny. There can be different stages, like there can be an error in the replication, there can be an error in the crossing over the sister chromatids, there can be a defect in the spindle formation, and there can be a defect in the cell cycle regulation points. All this results in the misaggregation of the chromosome, accounting for approximately 75%, and all this is associated with RPL. We have a different diagnostic strategy that we follow in recurrent pregnancy losses. It is basically molecular cytogenomics based, which basically is a refined amalgamation of conventional cytogenetics that deals with the karyotyping, as well as the molecular cytogenetics that encompasses of the fish and microarray. So just to give you a brief overview of what Dr. Ghosh has already told, is that the diagnostic strategy is bifurcated into two aspects. The one being if the couple, those are experiencing recurrent pregnancy losses that have come to us, and the other is the fetal as a specimen, the POC product. So if it is a case of the couple who are experiencing RPL or normal spontaneous pregnancy losses, so in that the couple is recommended for karyotyping. Karyotyping is basically a normal technique in which all the 46 chromosomes are aligned together, and we look for any kind of structural or the numerical abnormality that has occurred in the parents and that are affecting the normal conception or are leading to the normal or are leading to the pregnancy losses. And if the karyotyping comes out to be negative, the couple is then further recommended for further microarray analysis, which will be through which we'll be able to detect any kind of micro deletion or micro duplication that is suspected in the couple. And if we are screening for the product of conception, then the normal strategy is to go for karyotyping and to look for any kind of gross chromosomal abnormality that has occurred. If the karyotyping comes out to be normal, FISH can also be implemented. Like we can go for screening of the 5 standard chromosomal markers, that is chromosome 13, 18, 21, X and Y. And if both FISH and karyotyping are coming out to be normal, then in order to get an in-depth view of what the genome has occurred or what malfunctioning in the genome has occurred in terms of the chromosome deletion or duplication, then we can go for microarray testing. So karyotyping is basically the arrangement of the chromosome. We basically grow the fetal cells or the blood sample that is being obtained from the parents in a normal sterile environment. Once the cells are grown to the metaphase stage, they are then arrested and they're then harvested and followed by G banding. As you can see on your extreme left corner, it is basically the objective of 10x where a group of cells is seen. And when we use the same view, the view on the 100x magnifying objective, that is the oil immersion, then all the clearly well spread chromosomes can be easily seen, and they are very beautifully banded. And then all these metaphase spread of metaphases are then aligned together using a cytovision software, and the final picture that we obtained is known as a karyogram. The fetal chromosomal abnormalities basically occur because if there is any kind of chromosomal rearrangement that has taken place in the parents, and this is then forwarded to the further progeny. In the case of the reductional division, and it will further result in the abnormal fetal fetal development. So there are basically two kinds of chromosomal abnormalities that may occur. It can be a numerical aneuploidy, that is, it can be a gain of the complete chromosome, or it can be a loss of the chromosome, or it can be structural aneuploidies, as you can see in the diagram, that it accounts for approximately 3.1% in the case of karyotyping. And so structural aneuploidies are basically any rearrangement that has occurred in the segment of the chromosome. It can be an insertion, that is, a part of the chromosome is being inserted. It can be a deletion, a part of the chromosome is lost. It can be inversion, that is, a rearrangement has occurred in the same chromosome, or it can be a translocation, that the two chromosomes are involved and their segments are being exchanged. So it has been seen that normally in the gestation period of 6 to 18 weeks, the aneuploidy is very common, and in terms of trisomies are very common, accounting for 2 to 3% of the trisomies are normally associated with potential heritable translocation. Among these, triploidies and tetraploidies are also very common, indicating that there are more than one form of the haploid chromosome complement that is present. In triploidies, the common abortus fetus tissue have a karyotype of 69 XXY, or it can be 69 XXX. That indicates there is a dispermy. And if we eliminate all the causes of the maternal cell contamination, the true rate of mosaicism has been found to be 6%. This is basically a representation of how we do the processing at Dr. Lal Path Lab once the POC sample reaches us. It is the sample is then opened in a sterile environment, and from that the fetal tissue, specifically from the ribs and the skin and the villi sample can be taken for further analysis. It is then washed thoroughly, and then it is then incubated in the sterile environment under ambient conditions for the cells to grow, and the cells are then arrested and then harvested, and then finally, as you can see, these are then kept in the incubator, and then after harvesting we get the metaphases, and these are then further analyzed. The spectrum of the data that was generated from Dr. Lal Path Lab in the past few months has been compared with that of the US group of scientists, and we found that the most common aneuploidy that has occurred in the fetus is that of the trisomies, accounting for 9.89%, and it was found to be very well correlated with that of the data from the other lab. And the trisomy 16 was found to be the maximum, then followed by trisomy 21 and trisomy 13. We have also found few cases of monosomy X that was accounting to be 5.21, and triploidies and tetraploidies were also very well visualized. Like any technique, karyotyping has also a limitation. The karyotyping cannot detect, is not very useful in the case of molar pregnancy, as the karyotyping in case of molar pregnancy for a fetal product will come out to be normal, and the karyotyping has a limitation of detection of LOH, that is loss of heterozygosity regions. So in molar pregnancy, although the karyotyping is normal, and the LOH concentration would be very high. So that is one limitation. The second limitation of karyotyping is that as we all know, that the POC product is prone to contamination. So if it is a non-viable tissue, then the culture rate will be very high. If it is a case of if the fetal tissue is embedded in a paraffin block, then also the karyotyping is not possible. And further, if we get on the analysis of a POC product, we get a marker chromosome or any other additional material is visualized. So for in-depth characterization of that marker chromosome or the additional material, karyotyping has a limitation, because it has a limitation of detection of 5 to 10 MB. So for that, further analysis through microarray is very much recommended. So coming over to FISH, I'll be just touching this particular topic. FISH is basically the use of the fluorescently tagged probes that have complementary region to the gene of interest. So any commercial probe that is readily available is fluorescently tagged, and it is then found to then it is binded to the target region of interest. If that particular gene is present over there, then we'll see the signal. If that gene is deleted, then we won't see the signal. For aneuploidy detection detection of fish, of POC samples. We have a method through fish, and in this particular case, we see that all the 13, 18, 21 X and Y chromosome can be easily visualized. On your right hand corner, there is a fish picture in which the chromosome 21 was marked with orange color dye, and the chromosome 13 was marked with green color dye, and aqua is representing the chromosome 18. As you can clearly see that there are three signals for chromosome 21. It indicates that this particular sample is trisomy 21 positive, persistent with the Down syndrome. As we know that most of the genetic disorders are basically occur because of the DNA rearrangements, and these DNA rearrangements can be biallelic, multi-allelic in nature, can be because of the non-homologous chromosomes, and which basically further result in interstitial deletions, duplications, and inversions. So the most of the gross chromosomal abnormalities can be easily detected through karyotyping, but it has a limitation of detection of 5 to 10 MB, and the detection rate is only 3 to 5%. So in most of the cases, the etiology remains unclear. So for that, the copy number analysis becomes very important, and the copy number analysis can be done through chromosomal microarray. Copy number analysis basically refers to large regions of the genome that are either deleted or duplicated. It accounts for approximately 13% of the human genomic DNA, and CMA is basically, that is chromosomal microarray, is basically provides a high genome profile, high genome resolution of the whole complete genome. So either very small micro deletion or duplication can be easily detected, and it has a detection limit of to as low as 20 kb. Just to give you a brief, the microarray basically consists of 25 oligonucleotide probes. These have complementarity with the different regions of the gene of interest, and these are then through the photolithographic effect, these are spotted onto the glass surface. Microarray has the ability to detect any kind of aneuploidy, any kind of unbalanced translocation, LOH and UPDs, all can be detected through microarray, and even a very small micro deletion or duplication can be easily detected. And currently, we have three microarray platforms that we are using in Dr. Lal Path Labs. Not much related to this topic, but I would definitely want to throw some light on the ACMG and ACOG guidelines. That these basically recommend that any individual who is being suffering from intellectual disability, autism, or mental retardation, so they should be the first year line of test should be microarray for them, and for them we have the microarray 750K, HD, and Exon platforms that provide a very large coverage to detect any kind of abnormality. On the other hand, we also do microarray for all the prenatal samples. It can be amniotic fluid sample, it can be a cord blood or a CVS sample, for which the microarray platforms that are routinely used are Cytoscan, Optima, and 750K. For the products of conception, we get a huge amount of request, and for that particular specimen we use Cytoscan Optima platform. Cytoscan Optima platform has a very low coverage as compared to 750K and HD platform, but in case if we are using it for the BOC sample, then it has a set of 2.65 copy number markers. So any kind of chromosomal abnormality that is happening in the POC sample can be easily ruled out. So just to give you an overall picture of the capabilities of microarray, the first one is the microarray can detect any kind of copy number variation event that are occurring in the genome. It has the ability to detect any kind of breakpoint determination. Like in normal cytogenetics, we basically, it depends upon the expertise of the person who is analyzing a particular case, that what is the breakpoint where the chromosome breakpoint has occurred. But in microarray, it has the software itself gives us the exact position of where the break has occurred. So thirdly, the microarray also has the ability to detect the loss of heterozygosity regions. It also has the ability to rule out any kind of genomic contamination, and this becomes extremely useful in the case of the POC samples which are prone to maternal cell contamination. So the software has an inbuilt system to nullify the maternal cell contamination, and so what all we get is the completely pure data. Moreover, it has the ability to detect even a small kind of mosaicism also. Like the karyotyping, we have a limitation of the number of the metaphases that can be screened. In fish, we have limitation of the number of the cells that can be screened. But that thing is completely overpowered in the case of microarray, that even a small mosaicism copy number changes. It can be 1.2 also, so it can be easily detected through microarray. And microarray is also very well successful in the case of Mendelian consistency checking and also for trio analysis. So these are basically the three main pillars of cytogenetics. It is the karyotyping. The first picture is the karyotyping. It gives a whole genome profile of an individual, but at a low resolution. So any kind of numerical abnormality can be identified by karyotyping. But if it is smaller structural change that has occurred, it has a limitation over there. Secondly, the fluorescence in situ hybridization technique. In this, it is very useful technique if we are focusing on a particular gene or a particular target for which the probes are commercially available. But if we are but if we do not have a targeted gene, then in that particular case fish cannot be useful. So just to overpower both these techniques, microarray provides a complete resolution to any kind of micro deletion or micro duplication that has occurred in the genome. So any kind of copy number variation can be easily detected through microarray. So this is basically a comparative representation of all the cytogenetic tests that we have. As you can see, the resolution of the G-banded chromosome, that is the karyotyping, is 3 to 5 MB, and the metaphase fish has a detection limit of 100 kb. So any targeted gene who has a size of 100 kb can be easily detected, and SNP microarray can go as a detection limit can go to as low as 10 to 100 kb. The sensitivity is 10 to 15% in case of karyotyping, and fish it is 1 to 5%, whereas in microarray it is even more sensitive. It is 10 to 20%. The culturing, it is a drawback that we have to grow the culture. We have to grow the cells in culture, which basically delays the time and delays the assessment. So in karyotyping, the culturing is required. So the TAT increases. In fish, although there is no culturing, but it has a limitation in the terms of detection. But in microarray also there is no culturing is required. So microarray is a quick technique which provides you with complete information. If we see at the global perspective of it, the karyotyping definitely gives a complete profile of an individual, but it has a limitation of resolution. Fish is a very focused technique, so definitely the global perspective thing doesn't match to it. And microarray completely provides the information about the genome. Yes, microarray fish and karyotyping can detect all of them can detect the unbalanced translocation, and the other thing, the only limitation of microarray is that it cannot detect the balanced translocation, which can be detected through karyotyping. So and AOH regions or the LOH regions can be easily detected through microarray, which cannot be detected by any of the other two techniques. I have discussed about all the platforms of cytogenetics that are readily available for the cases of recurrent pregnancy losses and also for products of conception. Apart from that, I just want to give you a brief about the other factors, male infertility factors, that definitely play a very important role in RPL cases. Besides the basic evaluation of the male infertility factors, that is normal semen analysis or the biochemical test or the endocrinological testing, we also have a FISH and a PCR based testing testing method in order to see that whether is it the male who is responsible for recurrent pregnancy losses. So in fish we have two tests, that is, one is sperm aneuploidy detection test that basically goes in for screening of six marker chromosomes, that is 8, 13, 18, 21, 16 and X and Y. So all the chromosomes can be easily screened using the semen as a specimen. And the second method that we use is the sperm DNA fragmentation assay. It basically assesses the amount of fragmentation, DNA fragmentation that has occurred in the sperm sample that is resulting that is resulting in the failure of the normal conception or is resulting in RPL. The second strategy that we can use is the PCR-based method that involves the Y chromosome microdeletion test. It basically deciphers the intactness of the complete Y chromosome region, catering into all the AZF-C, AZF-A, and AZF-B regions. So this is just the diagrammatic representation of few of the cases that we have found, that in which the first one is basically the picture of the Y chromosome microdeletion test. In the yellow highlighted boxes basically represent the AZF-C region was found to be deleted in this particular male who was suspected with azoospermia. The second picture that you can see, the green signals basically indicate the amount of fragmentation that has taken place in the semen sample, and the more is the fragmentation index, the lesser are the chances of normal conception. Or so in this particular case, it was found to be 38%, which is pretty high. So in this particular case, the chances of normal conceptions are very limited, and the person definitely has to go for IVF or for a sperm donor. And the last case of the sperm aneuploidy detection through fish method, in which we have gone for the screening of the few marker chromosomes, and this particular case was found to be normal. So I'll be just requesting Dr. Saurabh to take forward this presentation and discuss few interesting cases related to RPL. Dr. Saurabh, thank you.
Thank you, Dr. Leena, for nice presentation and explaining all the aspects of recurrent pregnancy losses. Here, I'm taking you very quickly with few interesting case studies what we have experienced in our lab, which is very important to understand that how we detect abnormalities and how it is helping patients to manage their problem. So first case is rare complex translocation resulting in RPL. This is very interesting case because we found three-way translocation jumping between chromosome 1, 2, and 7. So the history is a couple married for 3 years with a history of 3 abortions. Wife is 31 years, suspected with secondary infertility. All the biochemical tests were normal. And husband was 32 years, seminogram revealed sperm count of 45 million per ml. So what we found, the left hand side, what you can see is a karyotype of wife, which is normal. You can see all 46 XX chromosomes are there intact, no problem. But husband shows this type of translocation, which is very, very rare. So what happened here is chromosome 1 is translocating into chromosome 2 is translocating to chromosome 7. And then abnormal chromosome 7 is again having a break in a telomeric region in the long arm and translocated to chromosome 1. So this is completely jumping translocation and very, very rare phenomena what we experience in our lab. And this is something which we wanted to publish, of course, with the consent of a patient. So what is happening here? So it is a rare double reciprocal translocations. So due to this translocation, the breakpoint in this rearrangement and the nature of the unbalanced derivative chromosome which could be inherited in the future pregnancy, the chances are having abnormal pregnancy is very high. So genetic counseling is of course very important here, because patients should be counseled properly that what abnormality you are carrying and what are the chances of getting abnormal pregnancies. Chromosome analysis of patients' first degree biological relative is also important to understand whether other carriers are also there in the family, where rearrangement are also present and who are at also in a high risk. Coming to a second interesting case, this is a case of double reciprocal translocation resulting in recurrent pregnancy loss. A couple married for 8 years with a history of recurrent pregnancy loss. And you can see in the left hand side, the husband is normal here, but wife is carrying again this type of rare translocation, double translocation. So what is happening here? Chromosome 11 showing arrow is translocated into chromosome 16, and chromosome 15 is translocated into chromosome 17, and the large portion of 17 is translocated to chromosome 15. So this is a separate dual translocation, what we have seen in our patient. Again, the same phenomena. So most likely, balanced translocation where two chromosomes have joined together results in no extra or missing chromosome material. That's why patients are, the couple or whoever is carrying this translocation is phenotypically normal. The joint or balanced translocation may not segregate appropriately during meiotic formation of gametes, producing a conceptus with unbalanced chromosome. So because of this unbalanced translocation or unbalanced gamete, the fetus is not able to survive, and they may miscarriage, and then we have to check for products of conception material to understand whether they are carrying that unbalanced translocation or not. But I want to point out here, sometimes it is very difficult to follow up the cases in a lab because we don't get products of conception every time, and we get parental karyotype, and after we follow up with the patient, we don't understand what is happening because they may not being able to send a sample to us again. They may send it to somewhere else. So that is very difficult to follow up cases sometimes. So the doctor should offer chromosome testing to couples with 2 or more miscarriages. Of course, it is very important now. Explaining the implications for explaining their infertility, pregnancy loss, for chromosome aberration in their current pregnancy. Additional consideration should also be observed because maternal causes of pregnancy loss like chronic disease or clotting abnormalities that can lead to antiphospholipid syndrome and maternal illness is also very high, which Dr. Ghosh has already explained. I don't want to spend time here. Couples with infertility and multiple miscarriages should also be referred for reproductive medicine, reproductive endocrinologist for examination of hormone deficiency. This is a case of products of conception. So this is the case which has come to us after pregnancy loss, and this tissue has been karyotyped, and what we have seen, all the chromosome were having extra copy. So usually we should see 2 copies of each chromosome. Here we are observing 1 extra copy of each chromosome, leading to 69XXX. This is a triploidy case. This patient is at very high risk for live-born offspring with congenital abnormalities and miscarriages. Genetic counseling is recommended, and prenatal diagnosis should be offered for future pregnancies. Chromosomal analysis of parents or first-degree biological relative is suggested to identify other carriers of this rearrangement who are also at risk. This is very interesting case because we have done FISH also in this case, and this is a case of prenatal diagnosis. So what we found that ultrasound findings for female with 37 years revealed multiple congenital anomalies during ultrasound. When we did karyotype and FISH, we found this abnormality which is called as trisomy 18 or Edwards syndrome. In FISH also we have seen 3 blue color signals. So these 3 blue color signals indicate that the patient or a fetus has trisomy 18, and unfortunately it is very, very lethal abnormality. So FISH analysis confirmed presence of trisomy 18 persistent with Edwards syndrome. The clinician should counsel the patient on the risk associated with the current pregnancy, and further couple karyotype can also be offered. Unlikely, it is the risk for getting trisomy 18 in next pregnancy is very low, but still prenatal diagnosis should be considered. This is a case of Joubert syndrome. So Joubert syndrome is rare in India, but we have found this case in our patient. The female was 36 years with 3 abortions. Chromosomal microarray was offered directly. So we didn't do any other tests like FISH or chromosome analysis or karyotyping. We did microarray in this patient in POC sample, and ultrasound was also suggestive of multiple congenital anomalies. What we found is a chromosome 12 deletion in the long arm. 12q21.32 is a location of chromosome 12, and we found this particular deletion here. So what is Joubert syndrome? Joubert syndrome is disorder of brain development that may affect many parts of the body. It is characterized by the absence or underdevelopment of cerebellar vermis and malformed brain stem. So we found this abnormality and has been reported. Genetic counseling was done, and all future pregnancies was advised to have a prenatal diagnosis. This is case number 6 with tetralogy of Fallot. So the patient has come to us with the tetralogy of Fallot. The POC sample has come to us. Female was 28 years old with recurrent abortion. fifth gravida and cystic hygroma was complained with the patient. And what we found is 15.5 MB deletion. So 15.5 megabase deletion is very, very large deletion found in chromosome 6q21. But this 6q21 is not only responsible for tetralogy failure. So when we did a literature review, what we found, there is a paper available here, chromosome 6q deletion report of a new case. And there was some review of literature also done. So tetralogy of Fallot was one of the characteristic feature of 6q deletion. That's what we have correlated here. And this is very rare chromosome abnormality detected by chromosomal microarray. And let me tell you that this particular deletion of 15.5 MB is also very difficult to see in chromosome analysis, in karyotyping. So here microarray is very powerful to detect this abnormality. Yes, so marker chromosome, what Dr. Lina was explaining, that sometimes, in karyotype, what we see is a marker chromosome. So what is marker chromosome? Marker chromosome is a small chromosome which is shown in this arrow. It looks like a dirt particle, but it is a small chromosome part which is lying in between a metaphase. And sometimes it is very, very difficult to understand from where it has originated. So this type of chromosome abnormality can be characterized by microarray very well. So here the history is female is 38 years with recurrent pregnancy losses, male partner carrying supernumerary marker chromosome. This is again interesting. Sometimes, most of the time what we see is supernumerary marker chromosome will be carried by a female partner. But in this case, we have seen that male partner has carried this supernumerary marker chromosome. When we did microarray, we found a gain of chromosome 9. So this is, although this is a marker chromosome and it is showing a duplication, so we can correlate that chromosome 9 has an extra copy of short arm. This P arm indicates a short arm, and that has been detected through microarray. And this chromosome abnormality was of 30,208 kilobase or 30.2 megabase size, which is very, very large according to microarray analysis, and that has been reported. Microarray is again very important to detect multiple abnormalities. That's what we have seen here. So in this particular case, we do not have much of history in this patient. And what we have found, in one sample, we have found trisomy 22. That means extra copy of chromosome 22 was discovered. Trisomy 7, extra copy of chromosome 7. And there were, again, extra chromosome abnormality. Lots of other chromosome abnormalities were seen. So this last circle shows that chromosome, right from chromosome 4 to different chromosome, has been observed. To conclude our talk, including Dr. Lina's and my case study, chromosome karyotyping in couple is extremely important in all the cases with recurrent pregnancy loss. So proper counseling and follow-up is extremely important, as a copy number change of unknown clinical significance can be identified, which is comprising of 5 to 10%. Microarray is very, very powerful technology with than karyotyping and FISH, because microarray can detect a smaller micro deletion or micro duplication or any other small supernumerary marker chromosome characterization or added material chromosome characterization. But karyotyping is also important because karyotyping is the only technology which can detect balance translocation, which we have seen in our case studies. It is now recommended that microarray should be offered in any pregnancy with ultrasound anomalies and a normal karyotype. So even if products of conception is sent to lab, it should be detected through chromosome microarray first, as Dr. Ghosh has also explained. So microarray can detect some abnormalities. If microarray is normal, if we don't find anything, then of course parental karyotype is important here. So here we end our talk here, and over to Sankalp.
Thanks a lot, Dr. Saurabh. So we have few queries. So one question is, can PCOD or PCOS lead to a recurrent pregnancy loss? Dr. Ghosh, if you could answer to this.
Yeah. Am I audible?
Yes, sir, you are audible.
Okay. Yeah, but studies have shown, and also our clinical experience has shown, that polycystic ovarian syndrome women have a problem in conception, and if they conceive, they also have a high chance of having pregnancy loss. Now, there are some interesting theories which have come up regarding the issue of recurrent pregnancy loss with polycystic ovarian syndrome, one of them being high LH hormone around the conception period, and also luteal phase defect, which is common in patients with recurrent pregnancy loss and polycystic ovarian syndrome. Now, apart from there are some interesting case reports which suggest that PCOS could be genetic in origin, though it has not been proven beyond doubt, and exact genes have not been identified. But this is something very interesting and would be a point for future research. Thank you.
So another question is that how we should proceed for resolving a RPL case, whether to do a karyotype or FISH or CMA, how to decide which platform will be the best for resolution.
Well, as far as, Saurabh, please go ahead.
Sir, please, if you can answer this.
Yeah. You see, traditionally we have been doing karyotype. That's what we have been taught, and guidelines also suggest karyotyping. That in product of conception, we do karyotyping of the product of conception. But recent literature, as far as it goes, and it's going towards, it is showing that probably chromosomal microarray would be the best means to interrogate product of conception. Saurabh.
Yeah, very rightly said, Dr. Ghosh, that traditionally karyotyping is being practiced from quite a long time, and karyotyping is also
Hello, can't hear.
If it has been discarded by mistake, then couples karyotyping is also important. But if products of conception is available, then it is very important to understand the chromosome etiology of products of conception by microarray, and if possible by FISH. And then if it is normal or abnormal, depending on the abnormality, we can go back and check parental karyotype.
Saurabh, if we are doing CMA in product of conception, is there a necessity for doing FISH?
Not necessary, sir. Chromosomal microarray is enough to do enough to understand the chromosome etiology. But sometimes if patient is not affording chromosomal microarray cost, then FISH is also working for major chromosome aneuploidies.
Another question that comes to my mind is, suppose we do a product of conception chromosomal microarray and it comes abnormal. Then next step, should we do karyotyping of the parents or should we doing CMA of the parents?
Sir, karyotyping of the parents, because balance translocation is important to understand. Correct. If it is there. If it is normal, then of course microarray can be done.
Correct. Thank you.
Thank you, sir.
Thanks a lot, Dr. Ghosh, Dr. Saurabh, and Dr. Leena. While we are coming to the end of this wonderful session we had from our esteemed doctors, I would like to take this opportunity to discuss a unique initiative by Genewise. Genewise is a digital academic platform to share, collaborate, discuss academic information related to the fields of genetics. This can be from oncology, neurogenomics, or tests related to pre- and postnatal genetic counseling. To associate with this initiative, I invite you all to share with us any clinical case studies, academic blogs, write-ups, video bites, which can help to gain more knowledge, and we are decoding the genetics. So we will be featuring these articles along with doctor's profile on wide digital network of Dr. Lal Path Labs. If anybody of you are interested, do write to us at genevolve@lalpathlabs.com. As far as accreditations are concerned, we are CAP certified and NABL certified with ISO certifications. So Genewise brings the dawn of new era of genomics testing. Thanks to all the speakers and participants. The recording of this session will be available on our LinkedIn and Facebook site. We will now close this session and look forward to many more such fruitful sessions. Thanks a lot, everyone. Thanks a lot, Dr. Ghosh.
Thank you. Thank you very much. Indeed, it was a pleasure.
Thanks a lot, sir. Thanks a lot. Thanks a lot, Dr. Saurabh.
Thank you. Bye.